Hi, everybody. Good morning, and thank you for joining us today on this Tuesday morning. Today we are getting a lot of people rolling in right now. So this is a Precisionary Webinar. We are hosting Dr. Brian Christie today. And it's my pleasure to introduce Professor Christie, who is a researcher in the field of brain plasticity in neuroscience. And we've known Dr. Christie for many, many years because he's used our compressed tone vibratone for his electrophysiology research. So Dr. Christie obtained his Ph.D. from the University of Otago and has since held positions at Baylor College of Medicine, the Salk Institute, and University of British Columbia. And currently, he leads the lab at the University of British Columbia Island Medical Program. He focuses on brain plasticity and recovery mechanisms following both acquired and congenital neuropathological conditions. And in today's webinar, Professor Christie will discuss the intricate realm of mild traumatic brain injury, or MTBI. With his expertise, he will analyze the clinical relevance of closed head impact models, talk to us about the brain's inflammatory response post mild TBI, and evaluate the impact of such injuries on critical brain structures like the hippocampus, which you can also see in the symbol for the Christie lab. Moreover, he's also going to share with us what he has found regarding sex-specific alterations in brain plasticity subsequent to mild traumatic brain injury. So join us for our session today as we look at mild TBI and explore avenues for enhanced functional recovery under Professor Christie's expert guidance. And with that, Brian, I want to hand it on over to you.
All right. Well, thank you so much for that wonderful introduction, Amy. Really appreciate the kind words. And for those of you joining in, the pictures at the top are located close to where my laboratory is situated in Victoria. And we're part of a distributed medical program from the University of British Columbia. So I have appointments at the University of Victoria under the Division of Medical Sciences, the University of British Columbia in Cellular and Physiological Sciences. And I'm also an adjunct professor at the Department of Psychology in San Diego. So we have a lot of collaborations and a lot of different laboratory space around. But this is by far, one of the most beautiful places to be situated. For those of you who are looking for future graduate or postdoctoral positions, we are going to be hiring in the coming year, and you're welcome to reach out to me afterwards and if you're interested. My laboratory in general is interested in neuroplasticity, and we use it as sort of a metric of network function, and this is related to today's talk. We have a history of looking at things like how exercise and diet can enhance learning and memory processes in the brain, whether that's a neurogenesis or a change in cell numbers or a change in synaptic strength. We also have a long history in looking at neurodevelopmental disorders like fetal alcohol spectrum disease or fetal spectrum disorders, and looking at different therapeutic options for those disorders. And lately we've expanded this into prenatal cannabis, and this is a rising issue in today's society, and particularly in Canada, where cannabis has been legalized. And so you have increasing numbers of women who are continuing to consume cannabis throughout pregnancy. And of course, today we're going to talk about our recent work in looking at traumatic brain injury. Now, this work is also featured on our new laboratory webpage, and I've put a link up for it in the chat. And my trainees, spent a lot of time creating little videos that actually show all of our protocols. So we've tried to keep them under 5 minutes. They're nice, short, and we also have graphical protocols that allow people to follow along all of the different methods that we use, and we show, different types of experiments, whether it's, using slicing protocols like the compressed-tone, or how to do different types of histology or in vitro electrophysiology. Always welcome any comments or suggestions you may have, or if you want any additional videos, always feel free to reach out. So Most of today's, we're going to start with looking at traumatic brain injury and the different types and how we can best model them experimentally. So here we have a series of pictures that actually represents different severities of traumatic brain injury. So on the left-hand side, we actually have just players engaged in sport. And you can see here on the left-hand side is actually a picture of my daughter playing hockey. And in hockey, soccer, football, basketball, you tend to have a variety of impacts that occur as you collide with other players or run into posts or things like that are just bumps, but they are actually subconcussive blows that actually exert physical forces upon the brain. Now, when those forces become a little bit greater, sometimes they can result in unconsciousness or sensory impairments. And these are mild traumatic brain injury and something that can be accompanied by a loss of consciousness. And typically this is for under 30 minutes. Now, as we start moving into the more severe types of injury, typically you have a loss of consciousness that's greater than 30 minutes for both moderate and severe TBI. And one of the other differences between moderate and severe TBI and mild or subconcussive injuries is that there's evidence of injury in CT scans. So in someone who has a moderate or severe TBI, whether it's from a stroke or a penetrating injury, you'll see bleeding in the brain and structural damage in the brain. Typically, you don't see this with mild forms of traumatic brain injury or subconcussive injuries. And it's important to recognize these differences because almost 85% of all TBIs are of this mild type. And the number of subconcussive injuries really hasn't been well documented because we're only starting to realize that participating in these contact sports can actually have transient effects on cognitive function. So how do we study TBI? Well, in the main part of the literature, people have been using more invasive models, and they on the left-hand side here we have a fluid percussion model, and so this is where you actually stick a tube through the skull of the brain, and you're going to allow fluid to access the brain and compress the tissue directly. And on the right, we have what's called a controlled cortical impactor model. And here again, you make a trephine hole, but a rod is going to penetrate and it's going to push down on the brain and it's going to deflect the cortices to a certain degree. And that's how you determine the severity of injury. And down below, we see some results of these types of injuries. And you can see whether it's mild is on the left or more severe on the right, there's always some structural damage. Now, in the human condition, this would already be considered a moderate injury or a severe injury on the right. These aren't really mild injuries like we're hoping to study. And the models are also constrained by some issues with how they're produced. You have to make an invasive craniotomy so that you can access the brain with both models. And this process in itself promotes inflammation and really doesn't model the human condition where you typically don't have an open head wound in a mild traumatic brain injury. Because they have these surgical interventions, these models also include anesthesia as a requirement. And this has, sort of conflicting confounds associated with it. In the first place, anesthesia can be neuroprotective, so you can have induced comas to help prevent, exacerbated activity with a traumatic brain injury. But when you're anesthetized, your ability to self-maintain body temperature is also poorly regulated. In animal models, this can result in the promotion of tau hyperphosphorylation. And so you're actually mimicking conditions that seem to be associated with artifacts that are close to what you would expect to see in things like Alzheimer's disease. So they're not the most ideal models. And a lot of the laboratories, and funnily enough, a lot of them have been concentrated in Canada, have started to look at different closed head models of a mild traumatic brain injury. In our laboratory, we've been working with an awake closed head injury, and we've been assisted by Dr. Sandy Schultz from Monash University in Australia. And I'll explain some of the contributions he's made to the model as we move along. And that the model has started to take off and proliferate in a number of countries. But there's also this pressure wave model that's been used in zebrafish that Ted Allison made in the University of Alberta. There's a lateral impact model that was pioneered by Rochelle Maciasiak at the University of Calgary. And I really like this model too, and we've been adopting it in our laboratory as well. I have a postdoctoral fellow, Eric Eolfsson, who started off in Dr. Maciasiak's laboratory and then moved to mine, and he helped introduce the model to us. And then there's also the Chimera model that was pioneered by Cheryl Wellington at the University of British Columbia. And the main difference between the model I'm going to show you today and these models is that the latter three all involve still the use of brief anesthesia. It's a transient, it's seconds to minutes worth of anesthesia, but they still require anesthesia for the models. The awake closed head injury I'm going to show you doesn't require anesthesia, and it's not really an impact in the traditional sense. So here's sort of the layout. We actually use a controlled cortical impactor device, and that's the device here in panel A. And normally this post would be used to depress the cortices after in a trephine hole, but instead we put a rubber tip on it, and then we 3D print these little helmets that we're going to put over the skull so that they're covering the impact zone. And here's an animal in a restraint cone, so it's an injection cone. They have the little helmet over their head to help center where we're going to put the probe, and it also distributes the force of the impact. And, typically when we think of impact, we think of something striking the brain or striking the skull at least, which is common in most injuries that we see in sports. But we actually liken this to more of a headbanger injury. And so here, if we watch the screen, you'll see that what we're going to do is we're going to lower the impactor to that helmet. And then rather than it hitting the head, it's actually rapidly accelerating the head forward. So you get all of the physical forces associated with a head injury, but without that striking. So we don't get bumps or bruises or anything. And when you remove the animals from these restraint cones, they're fairly normal. And that was actually one of our first challenges that we ran into, was figuring out how we could assess if we were inducing any sort of injury. Now, in the human population, you use something like the SCAT, or the Sport Concussion Assessment Tool, to assess cognitive function in the clinical population. Individuals, tend not to be able to do working memory tasks. They may have lost consciousness, and they may forget where they are and what they're doing at a specific time. And these are indications that you have actually suffered a mild traumatic brain injury. So for us, we had to adopt a novel procedure for animals, and typically with the open head or open wound concussions, one of the first things that people look at for a neurological assessment is the animal still breathing. When you're impacting the brain directly, often there's a cessation of breathing, and so you need to, hopefully reinstigate that. And then you want to look at how quickly the animals can right themselves. So when an animal's anesthetized, they typically will place them on their back and look at how long it takes for the animals to regain consciousness and right themselves. And so we incorporated that into our model. And then we also incorporated these sensory tests, because we end up finding that our animals don't lose consciousness very often. And so we started looking at sensory tasks like the startle response. And this is just a clapping noise that we do to startle the animal. In the worst conditions, if the animal's unconsciousness or it's transiently not aware of its surroundings, they have no reaction to that clapping sound. But most animals will respond quickly, have a whole body jump or a freezing motion to the start. Then we'll quickly transition to doing a limb extension. So we'll clap over the animals, then pick them up by their tail and hold them over a bar or the cage top to look at limb extension to see if they're aware of their sensory environment, have that proprioceptive awareness, and if they'll reach out and try to grasp what's in front of them. The absence of any sort of reaching movement is a 0, and a full extension and grasping onto a beam or bars of a cage is a full extension that gives you a full score. we'll then place them quickly on a beam. If we put them on the beam and they don't move at all, and sometimes animals that are stunned will kind of like swim and they'll be lying flat on the bar without actually grasping it. That would be a 0, and other animals will walk straight down it towards their home cage. If they are moving along the bar just fine, then we actually start rotating it. We're going to rotate it 4 times to see if the animals can follow along, kind of like a rotating beam that you might have for different types of motor tests. And we score it based upon how many times the animal can go around. So we're rotating it really slowly. If they make it around all 4 times, they get a perfect score. If they fall out on the first rotation, then that would be a score of 0. So we have a way of assessing their sensory capacity after a head injury as well. And with our protocol, what's interesting is that we typically don't see the animals as being unconscious. So that righting reflex is fairly rapid, and there's little delay in 90% of the animals that have a single injury. And with repeated injuries, and this is 8 times, so we go from 1 to 8 times in the repeated injury case, you see that there's little change in the degree of consciousness. So this is like, heading a soccer ball 8 times in a row, or having your body jolted 8 times in a row. But interestingly, when we look at those sensory tasks, we start to see a difference in what's going on. And so our sham animals, there will always be an animal that'll fall off the beam or not do exactly what we want. So we never have 100% in all 4 tasks. But with the single injury, we tend to see, 25% of the animals show some sort of deficits. And with the repeated measures, we tend to see about 50% of the animals show some deficit. But this is a short-lasting deficit. And at post-injury day 1, just a day later, we no longer see any of these deficits. So it's a quick, short-lasting deficit as far as behavior and sensory behavior goes. So when we started off, we talked about how we had these, gross morphological changes in the traditional models. And so one of my recent Ph.D. candidates, Dr. Hannah Reed, was interested in looking at the degree of damage that we had in our model. And so you can see in this model from the Linadol paper in 2015, when you do a mild fluid percussion injury, whether it's a very slight injury or you do it with increasing severity, you always get damage in the underlying cortex that can extend all the way down into the hippocampus. And here you see blood seeping into the ventricles and causing a shift in the hippocampus. And that damage is apparent in crescent-violent states. So Hannah looked at animals that were injured, either not injured, injured 1 time or injured 8 times. And this could be 8 times in a day. And you can see from the whole brain mounts that there is no signs of gross morphological damage anywhere on the cortical surface. And if you look at crescent-violet stains, and the arrow indicates where the impactor was placed to produce this rapid head acceleration, there's no sign of cortical injury in these animals, or at least gross structural changes in these animals. And this was a unique finding for us, because if you think about the human condition, and even on the CDC website, they tend to talk about this coup contre coup injury model, where they show the brain kind of rattling around in the skull, and you'll have the coup injury at the front of the brain where the initial contact is, and then you'll have the contre coup injury at the back of the brain when the body is recoiling from the physical assault, or the physical forces exerted on it, and that causes the brain to kind of rattle around like a pea in the pod. Well, those of us who are neuroscientists know that the brain is really tightly, wrapped in by the brain. It's covered by the meninges. The fluid-filled space is really sort of a tight protective packing measure, and the brain doesn't tend to rattle around like this. So we started thinking about how we could model, or what we could look for in our histology to see whether this coup contre coup injury model was right, or maybe if a different model was more appropriate. And so a coup contre coup injury model would have, in a rodent, would have these damage sites in the cortex around where the acceleration starts, and at the bottom of the brain for the contre-coup injury. In contrast, if we had more of a diffuse injury, your TBI event would cause the center of the brain to kind of move around and pull and stretch on surrounding tissue. So you'd see more of an injury that's in around the middle of the brain as pulling on the brain. And of course, the hippocampi in the rodent brain would be around this area, and they wouldn't be damaged as much in the coup-contre-coup injury model by ours. And so to look at this, we had a master's student, Katie Neal, who was actually working with Dr. Reed. And she decided to look at BRDU staining. And most of you might be familiar with this as a technique in which you can look at adult neurogenesis and cell proliferation in the brain. And so here we can see the cell proliferation in the dentate gyrus, clearly indicated here in these red circles. There's also some proliferation around the ventricles, going up the rostral migratory path. There's cells moving along, but they're very localized, discrete places that we expect to find cell proliferation. But when you look at an injury model, there's a lot of cell proliferation that's not constrained to these normal areas where we expect neurogenic growth. And if we expand that picture up a little bit, we can see that this cell proliferation is occurring well outside of the dentate granule zone area and extending throughout the hilus, the CA1 region, and up into the corpus callosum. But you don't see a lot up in the cortex, and you do see more in the thalamic regions of the brain. So it's more of this center or diffuse injury model. Now, Katie also did some secondary stains to try and identify what types of cells these were. And so she looked at her BRDU stains, and then she also did staining for microglia cells with IBA1 and astrocytes with GFAP. And when you do the merge data, you tend to find that the cells that are reacting or have BRDU present are cells that are activated microglia. And so these can be amoeboid shaped or reactive microglia that have their processes extending towards something. And so identifying what that something was the project of another PhD student, Juan Trevino Paredes. And he thought that the microglia activation may reflect small microvascular damage. And so he did this project where he started looking throughout the brain for tiny vessels that could be damaged by this, remember, quite mild injury. It's mainly an acceleration of the head up and down. And he was able to find both what he calls large vessels. So these are tens of microns large, and smaller capillaries, a few microns in size, where he could see obvious breaks. And so he would stain these with lectin in red. And then he would look for blood products. And one of the blood products we look for is fibrinogen. This is a clotting factor that's in the inside vessels normally, and not typically apparent outside of the vasculature in the brain. When it is apparent outside of the vasculature, it can activate CD4 receptors on IBA1 or microglia cells, and this can cause them to release inflammatory cytokines. So he looked for evidence of this, and he was able to find broken vessels with this leaked fibrinogen. And around the fibrinogen, he was able to show that there are microglia interacting with the fibrinogen. And so this is whether you have large vessels or on small vessels. You can see again these leaked fibrinogen, kind of like the vessel had been stretched and it caused a transient increase in porosity in this case. And again, you have microglia that are going within the constraints of the corpus callosum, which is a very dense, compressed, myelinated tract. These microglia are trying to move towards the fibrinogen, where they can interact and help clean up the detritus. A better shot of this comes from this one set he has from the thalamus, where he's got a few microbleeds, and you can show the fibrinogen leaked out. And then here, if we look in, we'll see a variety of types of microglia. First, there is this ramified or normal microglia that has all of its feet out, surveilling the surrounding area, looking for something that it needs to do. The activated microglia have these ramified dendrites. This is reaching towards this fibrinogen leak in the brain. And what was really interesting for us is we also found evidence for rod microglia around fibrinogen. So this is when microglia line up in these distinct rod-like shapes. And this is something that's normally associated with the Alzheimer's disease model. So we were really interested to observe this around the microbleeds. And then, of course, there's also these phagocytic or amoeboid microglia that are actually enveloping the fibrogen itself. So we're able to show that you can get all of these activation patterns and the phagocytic cells around microglia that are fixed in time. And this is shortly after the TBI, so that the bleed has occurred. We've got fibrinogen in the extracellular space, and microglia are looking at it. So we think, oh, this is great. We've got some evidence that these small microbleeds might be doing a lot. And so one spent a considerable amount of time trying to quantify the number of bleeds that he saw. And what he ended up finding wasn't exactly what we hoped to see, but it was that the microbleeds are distributed throughout the brain. They're a little bit focused more in the center than in the periphery of the brain, but there were some, and this is actually in the opposite side from where the impactor is pushing down. So it's pushing down here. So the brain should be more twisted on this side, on that side, but you can see lots of microbleeds in the center of the brain rather than around the peripheries. So what could those microbleeds and the microglia be doing? Well, one of our ideas was that this could be a way in which activated microglia start doing erroneous tragocytosis, and that's taking away or pruning spines from neurons and changing their ability to interact with other cells. And so to study this, we ended up needing to think of a new technique, because we couldn't, we were working with a rat model, so we didn't have inherently fluorescent cells. And we ended up working with another fellow at the institution, Dr. Patrick Nahirny, who was interested in working on this dye-eye technique. And so Juan and Dr. Nahirny ended up taking little specks of dye-eye, so in powder form, and so they get tiny, minute particles of it. They would actually crush it up further, the powder that you get in a pedestal, and then they would get one single particle on the tips of these sharpened tungsten needles, and they would place them all around, in this case, in the hippocampus. And you can kind of see in this picture, Juan has placed several pieces in the dentate granule cell, where we saw all those changes in cell proliferation. He's also placed some in the stratum pyramidale region, the CA1, and you can see tiny little dots of the dye-eye placed all over the brain. And what's really amazing about dye-eye is it'll perfuse along the cell membrane and it fluoresces. And so you get these brilliant pictures of individual cells, but only if you have these tiny little particles. If you have too big a particle, you get big, large, bright spots of staining. So you really have to have the tiny particles to have it trace individual cells. And we found it was most effective if you did it in the cell body layer. And then you can go down and you can start quantifying individual spines. Of course, this is quite a large task. Oh, I see it. We have a question about the baseline levels of microbleeds, and yeah, that was a question we were interested in, and they were very low in the sham animals. So you might find 3 or 4 within a brain. We're not sure if that was due to the perfusion procedure itself, or that was just a natural rate of occurrence. But there were, 4 to 5 times as many in the TBI brain. So we do think it was in relationship to the physical forces being placed upon the brain. Again, we have a very mild injury pattern. If you did this with a fluid percussion or CCI injury, you'd see significantly more microbleeds throughout the brain, because the brain's physically compacted by the probes. So So while we're able to visualize all these spines, of course, it's quite a procedure to count them. And one was grateful to have the assistance of two undergraduates in the laboratory, Emily Bostashen and Annika Willoughby, who were both NSERC undergraduate students in the laboratory. And they actually went through and counted spine numbers in different parts of the brain. And what we found was that there was a difference in spine loss in the cornu humanus versus the dentate gyrus of the hippocampus. In the cornu humanus, surprisingly, we didn't see a lot of spine loss. But in the dentate gyrus, right next to that ventricle, where you might have more of the pressure waves with these physical forces, we did see spine loss on these granule cells. And it was in both the ipsilateral and contralateral side to where the impactor probe was placed. And we were really, interested and worried in the beginning that we might see differences depending upon the brain actually moving sideways due to that probe rapidly accelerating the head, because you get a little bit of a twisting place, and that gives a little bit of shearing force that might be more on one side of the brain than the other. So we've always, tracked whether we're ipsilateral or contralateral to the injury site, and we've actually never seen an ipsilateral effect to the injury to where the probe is. They went on to also look at if there were changes in dendritic processes in these neurons, and they did a shoal analysis where you take concentric circles and place them around cells and look at the number of branches that you have as you move outwards, and also the total dendritic length as you move outwards. And while they found out that the average dendritic length within the shoal circles didn't change, the total dendritic length for a cell was decreased after TBI, and the branch numbers were also decreased after TBI. So the cells were actually retracting some of their dendrites and having less dendritic length and fewer spines on them. So this, of course, can have functional implications for synaptic plasticity, which is a main area of endeavor in our laboratory. And so this is where Dr. Eric Yulson came to the laboratory and started looking at the functional implications for a loss of spines. And he was using the compressome to make transverse slices. And for those of you who normally make coronal slices, to make a transverse slice, basically what we do is we have the brain is split in half. We put the medial surface down flat on, in this case, a petri dish on ice, and then we're going to make an incision along this dorsal surface to give sort of a gluing surface, so that we can have the dorsal surface down and the ventral surface up. We put that in the compressome tube, and then we can encase it in the agar and then make our slices. And that generates these really nice hippocampal slices that have dendrites that stay in the plane of observation. So if we want to fill cells with fluorescence to do calcium imaging or do whole cell and do reconstruction of neurons afterwards, we don't have cells that have their dendrites coming up and they're cut off as we move along. So what did Eric do? Well, he did a form of electrophysiology called field electrophysiology. And I know some individuals may not be familiar with electrophysiology, so I thought I'd just describe the technique briefly. On the right-hand side picture, you can see that there's this translucent pipette tip. This is a recording electrode. And then there's this dark concentric bipolar stimulating electrode. And they're both located in the medial aspect of the perforant path of the dentate gyrus. This is that area where we saw a reduction of spines and a reduction in dendritic length following traumatic brain injury. And so if you stimulate with this stimulating electrode and record here with the recording electrode, you'll get a stereotypic waveform that's composed of the stim artifact. So this is the conduction of electricity through the ACSF medium. And then that electricity is also going to go through axons, and this is called the fiber volley, the first little peak. And this is as electricity flows past the tip of the electrode through the axons. And some of those axons are going to release neurotransmitter. And that neurotransmitter is going to activate postsynaptic receptors, and you'll get this postsynaptic excitatory response that's mostly carried by AMPA receptors in the initial phase, and as you move out, NMDA receptors are activated after you have the depolarization necessary to alleviate the NMDA blockade and allow the NMDA receptors to be activated. And when we're measuring these, we're going to actually measure this initial slope of the EPSP as an indication of how strong our synaptic communication is. When you look at this on a graph, we can look at the response size on one axis and time on the other. And if you evoke these responses, initially it's very easy to get reliably the same size of response, so the same synaptic strength. And this is a requirement for all of our recordings, is that you maintain stability in your initial responses. You don't want a response that's changing in size, getting bigger or getting smaller over the course of this initial time period, because that lack of stability really makes it hard to tell what happens if you now apply a conditioning stimulus. And so after this period, we can give high frequency stimuli, like 100 hertz stimuli for 1 second, and this can increase the size of the responses. And this is also known as long-term potentiation of excitatory synaptic transmission, or LTP. And in the studies I'm going to show you in a second, we use 100 hertz for 1 second as our LTP induction stimuli. And there's a variety of patterns that you can use, but they always involve more of a high frequency stimuli to increase the size of the waveform and increase the capacity for communication. Now, you can also use low frequency stimuli, and here I'm showing 1 hertz for 15 minutes. And if you apply this stimuli, you actually decrease the size of the response or produce long-term depression of synaptic efficacy. So these are bidirectional ways in which we can control the ability of neurons to talk or communicate with one another. And this is how you can build networks of cells that represent different objects or situations for learning and memory processes. And so together, long-term potentiation and long-term depression are the most relevant biological mechanisms for learning and memory processes that we know of currently. There are other theories out there, but these are the ones that seem to be most reliably associated with learning and memory processes in the brain. And of course, we expect that traumatic brain injury, even mild traumatic brain injury, can alter these processes. And so this is what Eric started looking at. And so here in this picture, we can see the result of his initial experiments looking at adolescent brains. And we looked at adolescents because youth tend to be more highly engaged in sporting activities where they can incur different types of injuries. And so here you see that the sham individuals, had about a 60% LTP in response to that conditioning stimulation that we had. And here on the right with these graphs, he's showing STP, or the short-term potentiation, right after the application of the conditioning stimuli, and then long-term potentiation, or LTP, which is 60 minutes after. And we go 60 minutes because this is when you start getting into a protein synthesis dependent form of LTP. And what he found, surprisingly, with these adolescent brains is that, STP wasn't altered immediately after, so this is post-injury day 7. The amount of LTP was significantly reduced in these adolescent animals. And if you waited 30 days, so this is when those behavioral signs had changed, he actually found he had less STP, so the brain wasn't as activated by the stimuli. And again, you weren't able to sustain significant LTP in these animals. So this is actually quite surprising to us that the nature of the injury lasted a lot longer than we thought it would. And so now we've got some biological mechanisms we can, start looking at and start examining different molecular cascades that could be altered. Now, because we were doing this 30 days later in these animals, this is actually when the animals become adults. And, so they're being injured around postnatal day 30, and now they're 60 days of age, which in rodent lifespans is a young adult. So he also looked at whether you gave the injury to young adults if you get similar effects. And this work's actually just started, but he's already shown at the sham animals have, nice, robust long-term potentiation and short-term potentiation. But at post-injury day 7, these animals already show less STP, so there's a significant decrease in their ability to show a change in synaptic strength immediately after the conditioning stimuli, and they also have less capacity to sustain long-term potentiation. And again, he's showing here in his cumulative graphs every single slice that he's run and the amount of LTP and LTD that he's showing. So there's more slices that are showing less LTP in the TBI group than in the sham or control groups. Now, just to finish things off, we've also done some work looking at long-term depression. This is very preliminary work, but we have our sham animals in dark here, where we've looked at the equivalent of post-injury day 1 and 7 in animals that haven't received any sort of injury. And then we've looked at post-injury day 1 and 7 in animals that have received an injury. And we were rather surprised to find that we saw a deficit in our ability to induce LTD in post-injury day 1. This quickly recovered by post-injury day 7, in contrast to what we saw with LTP. In the LTP animals, remember, we saw this deficit at post-injury day 7, and back in the adolescent animals, we saw that it extended out to post-injury day 30. So with LTD, there's a different phenotype to the injury. There's a deficit early on, so we have decreased bilateral capacity immediately after the injury. And then there seems to be a shift where you have less capacity to induce an increase in synaptic communication following the injury. All right, so I'll try and bring it all together for everyone now. So what we've shown today in the talk is that this awake close hit injury can produce reliable acute neurological deficits. We predominantly seem to see vestibular and motor deficits right after the injury. We can administer this awake close hit injury repeatedly without showing gross structural damage. And at a lot of our injuries, we are doing 8 to kind of replicate that subconcussive injury model where for individuals who are engaged in contact sports. And what's interesting is if you search the literature, you can find that like individuals that have rugby practices or football practices or hockey practices actually do poorer on learning and memory tasks right after a practice than they do 3 days later. So there is evidence that there is a change in your cognitive function after any sort of head impacts. We show that there is evidence for microbleeds and microglia activation. We're really not sure if this is the definitive cause of some of the other changes we see, but there is evidence for it at least. And we do are aware that fibrinogen can trigger microglia activation. Our spine analysis shows that there's changes in the numbers of spines after repeated mild traumatic brain injury, and these are specific to the dentate gyrus. And there also appear to be deficits in hippocampal dendritic structure after the injury. And finally, the RN-TBI impairs hippocampal synapses' ability to sustain LTP and LTD. And this appears to be long-lasting in the case of LTP and maybe shorter lasting in the case of LTD. I see there's some questions coming up. Before I get to those, I'll just thank all the fabulous trainees I've had in my laboratory over the course of these studies. Here's a picture of us at the last Society for Neuroscience meeting that we attended, and a annual laboratory picture in the campus here. And of course, I need to thank all the funding agencies who have assisted in providing us with funding, and all the trainees with funding for performing their work. It's truly enjoyable to work with these individuals. And I'd also like to thank you all for taking the time to listen to me, and we'll go to questions now. Thank you.
Wonderful. Thank you so much, Dr. Christie. So we actually, as you mentioned, we have several questions already in the chat box. And what I like to do is try to make it interactive. So I'm going to start with John Giesler. If you wanted to unmute yourself, but go ahead and please ask your question.
There I am.
Hello.
Yeah, so I'm at Mitochond Pharmaceuticals, and so we're always interested in all things mitochondria. So my question was related to what have you looked at related to the mitochondrial calcium overload, free radicals, anything that related to the threatened tissue, the penumbra, ATP production. Have you looked at any of these things post and prior and after injury?
Actually, we haven't. I do have some anecdotal data about that, but a lot of our effort in the first years has been establishing the model and actually showing that it is a good model for mild traumatic brain injury, and that we do have these deficits that are persistent. And one piece of anecdotal data is that we actually see more persistent deficits behaviorally in only about 20% of the animals. And so to quantify that, we really have to run up our animal numbers, not to the point that we have yet. But that sort of goes along with the clinical populations. About 20% of clinical MTBI cases show persistent deficits. But the anecdotal data for mitochondria is we have a couple of EM experts who have been working with us, Patrick Nahirny and Pedro Grandes in Spain, and they've noticed that mitochondria tend to be displaced from around synapses in the images that they have taken so far, but we haven't quantified that. And of course that, I think is something that a lot of people have thought about for TBI injuries, that it is a metabolic crisis that's ongoing following the injury, and that could explain some of these deficits in spines and synaptic transmission. But we really haven't delved into that. Be happy to talk with you further and see if you have any suggestions for our work, or if you're interested in having any samples from our work as well.
Thank you. Yeah, we work a lot with Patrick Sullivan, Brad Hubbard, and blunt force trauma, repetitive blast, like the people in Ukraine pulling the howitzer. And then this falls into an area that's a little bit tricky because it's a lot of the kids recover naturally with time. And so it falls into a gray zone of whether or not the pharmacology of our mitochondrial uncouplers are working because of the pharmacology versus just the time of recovery.
Yeah, well, and now it's becoming more recognized that, especially in the adolescent population, that these even mild injuries may have more persistent effects on their learning capacity, and then people are starting to extrapolate from that maybe it's impacting their developmental capacity during these adolescent periods, by reducing learning and memory processes. So that's I think one of the driving forces we had in the model. My kids all chose contact sports, hockey, lacrosse, so there was a little bit of innate worry on my own part, about how this is going to impact them. Yeah, so we were always interested in these issues.
Thank you.
Yeah, no problem. Thank you.
Thank you for a great question. Next up is Dr. Yi Hua. Hi there.
Yeah, hi, Dr. Christie. So let's talk. So my question today is, so when you design your TBI experiments, so how did you determine like the number of impacts and also the interval like between them? Because I'm worried that, for example, if I is new to this area, so is it possible I did not apply sufficient number of impacts, so it did not induce like the injury to the animal at all?
Yeah, and well, so we went at that a couple of different ways. One is the neurological assessment protocol, that allowed us to say that there's a behavioral effect. But we've also been, doing some work looking at cytokine production in the brain and the periphery, and seeing how that changes over time. And there's a sort of a sequential, change in different cytokines. They tend to peak about 3 days after an initial injury. And so our initial interest was the repeated injuries and whether there was a difference between if you separated them by, a period of days, or if you concentrated them all into one day. So the idea of concentrating them came from the idea that subconcussive injuries can occur in any practice, any sport. And our injury model seemed to be so mild that it might mimic subconcussive injuries. So we actually have some studies where we've done 8 injuries within a 12-hour period in a day. So we sort of do it once every 1 to 2 hours. And then we've also spread them out across days to look at the injury. And we've done it on daily intervals, 2-day intervals, 4-day intervals, to look and see if there's a critical period where you may have an accentuation of the injury because the second injury or the third injury comes at a time point when you have inflammation already ongoing in the brain, and that could cause that runaway inflammation that seems to be associated with second impact syndrome or CTE, type syndromes. And we have, I think, a little bit of evidence for that, that if you give injuries 2 days apart, maybe a little bit a stronger injury than a milder injury, you tend to have more persistent effects with synaptic plasticity. And that's some work that Dr. Olson's currently engaged in, using the lateral impact model. And that's kind of like a sideways car accident model. We're going to have a video of that, I believe, available on our webpage fairly soon. We're being very conscious about what we put on the webpage in terms of showing animals in research use, etc. So, but we think that the looking at the inflammation pattern is a good way to justify when you're giving those additional impacts, right? You want to sort of see if you can hit that second impact syndrome.
Thank you.
Welcome.
Wonderful. And Dominic had already asked a question earlier, which I think you answered, Brian.
Yeah.
He had asked about the baseline levels of microbleeds. He had a couple of other questions. And in the interest of time, Dominic, did you want to unmute yourself and ask? Oh, you already unmuted. Great. Go for it.
Yeah, so I was just curious because I'm about to start experiments that look very similar to what you have. So I was just curious whether this spine analysis that you did, whether this was in a specific part and whether there is a specific part of the brain or the hippocampal circuitry where you saw most of the impact or the change. I think you might have mentioned, maybe I got it wrong, or maybe you can help clarify. There was the CA1, I think, is where you did some analysis. And then I thought I had you mention again that you did some analysis in the dentate. But then I wasn't sure if this is the same area where you were doing the field potential recordings from. And so I was kind of not very sure about the analysis.
Not a problem.
Me in the direction of where you most.
Yeah. So we did do spine analysis in both the dentate and the CA1. And what we found was that the spines were more reduced in the dentate than the CA1. And so that drove our initial studies on field recordings to the dentate first, because that's where we saw these initial changes. We haven't really looked at CA3 terminals at all. And that's an interesting point, because those big mossy fiber terminals may be more prone to the disruption by physical forces. So it's a really interesting study. We've actually got tons of tissue as well. So there's always the capacity to look at things like that, and we're always happy to collaborate. If that's an area of expertise and interest for you, we're more than happy to share any results or samples that we might have already generated to help facilitate any work you're doing.
Oh yeah, excellent. Thank you very much. I'm just kind of starting into this field, so right.
Yeah. Well, good luck. It's a lot of fun. Always happy to help any, when he's starting out in it.
I might be reaching out to your lab soon. Thank you very much.
Oh, you're welcome.
Wonderful. We had a raised hand from Dr. Baruch Campos. Go ahead.
Yes, great talk. So I was wondering if for the part of the your electrophysiology results were done on the hippocampal area, right?
Yes.
I wonder if the same results could be persistent in a different area, like the cortex or any other different area. And also, a second part of the question is that this is all been done in vitro. Have you ever thought about doing some in vivo longitudinal studies, like doing another imaging technique, like, I don't know, in vivo 2-photon calcium imaging, neuropixels, etc.?
Yeah, and well, we've thought about two different types of in vivo. In fact, we've done some with in vivo recordings from the hippocampus, so field electrophysiology again in the intact animal. I do always worry about when you make slices, you're doing the most severe damage you can to the brain because you're hacking it up. Still, it gives us differences between the injury models. So that was reassuring. In vivo recordings, your N is 1, whereas you can have multiple areas. You can look at prefrontal and hippocampus and thalamus if we wanted from slices from any one animal and look at multiple brain regions, which is, I think, one of the advantages of in vitro, and also really being able to visually identify the placement of your electrodes is another strength of in vitro. But I do agree that there are some advantages of going to the in vivo models. We haven't done the 2-photon experiments, and even though we have laboratories here that do a lot of 2-photon work and work with stroke models. There's Craig Brown is in the building. But one of the issues is they tend to use trephine windows that they attach to the skull. So they go through this laborious surgical process to make a trephine window. They attach it to the skull. And we were worried that even the rapid acceleration deceleration might produce an injury that's the result of the window shifting or moving, or that change in the pressurization of the skull that takes place when you remove part of it and place a window over it. So we have been a little hesitant in going forward with that because we weren't sure if it was going to be an accurate model, and also because it's such an involved process to do all of that work and put it through an animal care process to do an awake close head injury in an animal with a trephine window. So those are just some of the practical considerations that we are trying to navigate through as we think about the work. Again, this is all still fairly new, the model and our incorporation and what we can do with it. So we're more than happy to share our techniques and protocols with anyone who is interested. We're trying to make everything accessible also by having that web page up that everyone is welcome to access. And of course, if you have any further questions or want to see anything, feel free to write. We have set up the model in laboratories around the world now, and have helped people get going. And Ian Stewart in Australia just got one going. Sandy, of course, has one going, and I think there's a few other individuals in the US that are interested in it as well. So yeah, so hopefully it takes off.
Great talk and really interesting results. Thank you very much.
Great, thank you.
Thank you. Well, I just wanted to mention that Professor Christie's talk in our Q&A session will be posted as a recording to our website. And I'll also follow up with a link to everybody here so you can access it easily. And so with that, I want to thank you, Brian, so much. We hope to have you back and maybe even visit you up in British Columbia. So thank you so much. And everybody, thank you also for attending.
All right. Thanks, everyone. Appreciate it.